experimental design. So first, why are we doing experiments in statistics? Well, an observational study, as we learned about in the last section, observes individuals and measures variables of interest, but it doesn't make any attempt to influence the responses. Sometimes that's enough. A lot of times it's not. An experiment deliberately imposes a treatment on an individual to measure their responses. So if we want to know something very specific about a group or a sample or a population, we can perform an experiment, and that should be able to tell us something very specific. Now, the trouble with observational studies that is avoided when we do experiments is this problem of confounding. So confounding is when you have two variables that are so strongly associated that both of them could be affecting a response variable, and you won't be able to tell which one's doing what. If you have a well-designed experiment, then what you've done is you've taken steps to avoid confounding. You've gotten rid of all possibilities, not all possibilities, but most of the possibilities of any confounding going on. Observational studies fail at this because they're not actually interacting with any of the experimental units. It's for this reason that when we can, we should try to use an experiment. Now, when we're talking about experiments in sport, we know what the terminology is, right? So we have treatments. Treatments are a specific condition that's applied to individuals in an experiment. It can be a combination of specific values of a variable. If there are several explanatory variables, it can be a lot of things, right? But the treatment is whatever you're doing to the X variable, if you will. It's whatever you're doing to the experimental unit. An experimental unit is the smallest collection of individuals for which a treatment can be applied, right? So if you're doing an experiment to see how certain water molecules react some treatment, then the experimental unit would be a water molecule, right? If you're trying to do an experiment to see how people react when they are asked a certain question, then your experimental unit is one individual person. And when it comes to people, instead of calling your people in your survey an experimental unit, we typically will just refer to those experimental units as subjects. It's a lot more humane sounding, right? Now, the way you experiment in statistics is not the same the way you would experiment in science, right? In a science lab, you have your experimental units, you perform some treatment, and then you measure a response. It's a very basic A, B, C scenario, right? In statistics, we don't do that. In statistics, in order to determine if there is an association between the experimental unit and the treatment, to see if the treatment actually does what we think it may or may not do, we have to use random assignment to split them into two groups, sometimes more groups. So what happens is we'll take our experimental units, we'll use random assignment, so literally some form of randomness to assign them to one group or the other, and there are many, many ways to do this. And then one group will get one treatment, and the other group will get the other treatment. And then we compare the results. That is a statistical experiment. And of course, there can be more, and a lot of times you'll have a control group that receives no treatment or an inactive treatment, like a placebo is another terminology. So, in an experimental design, there are things that need to happen in order for your experiment to be considered. So first, there has to be comparison. You have to have a design that compares two or more treatments, right? Here we have two treatments being compared. Up here in a standard lab setting, that's not necessarily the case. In a perfect lab setting, I'm sure, I don't have a science degree, I have a math degree, so I'm sure scientists like to compare things. But in statistics, we demand that there is something comparative about our experiments, right? We have to have random assignment. You have to use some method that uses chance principles to decide what non-negotiable. Doing so helps create roughly equivalent groups. There are ways you can randomly assign units to groups that does keep them very equivalent, so that there's the same number in each group. And it also helps balancing the effects of other possibly confounding variables, right? If you're using a random assignment to sort from one group into the other, then if there's anything underlying that is having an effect, then we can get rid of that potential effect on our treatment, right? All right, so control. You have to have control. You have to keep other variables that might affect the response the same for all the groups, right? You can't have other variables from outside interfering in your experiment. Otherwise, who's to say if your treatment is actually the thing that's causing the response? You can literally have a control group as something you can compare it to if you like, and that can have an effect. On the test that you took on Tuesday, or no, Tuesday last week, Wednesday and Thursday of last week, one of the questions involved kids who watched different snack commercials and what they had. You'll remember, hopefully you'll remember, one of those groups didn't watch any commercials. That would be an example of a control group. They showed one commercial to one group, another commercial to another group, and the third group, the control group, saw no commercial, and then they measured what snack the kids picked based on what commercial they watched versus no commercial. So that is an example of a control group. And lastly, there has to be replication. You have to use enough experimental units in each group so that the differences in the effects on the treatments can be distinguished for chance, pure probability, like little differences, little nuances between the units, right? So ideally, you want as many as you can get. Smaller experimental groups, less reliable results. The more you have, the better your results will be. And ideally, in replication, you also want other people to be able to replicate your experiment. This is sort of not really statistical, but scientifically speaking, you want other people to be able to replicate your experiment and also get similar results. Otherwise, your experiment comes into question. People question whether or not you're being honest. Now, there are some good principles that we like to have whenever we can. First, blinding. When you have a blinded experiment, a single blind experiment, then the subjects or the experimental units do not know what treatment they received, right? So if I'm testing a medication and seeing its effects, then I can have subjects. I want one group to receive one medication, one group to see another medication or possibly a placebo medication, and they don't know which one's which, but I do. That would be an example of a blind experiment. They have no idea what they're doing, but that helps me keep track of what the actual effects of my medication are, right? These people from if I double blind an experiment, then that means there's another layer to the blind. Somebody sorts all of the medications for me, and I don't know, so puts them into a bunch of cups, and I don't know which medication goes into what cups. And then I go give the medications to the subjects that they're supposed to receive. The person at the back knows. I don't know who got what, and the people receiving don't know who got what. So subject nor the person interacting with the subject has any idea who's getting what. This is done in instances where it might be possible that my interaction, my expectations for what I would see could be swayed. And, that makes sense. If I am the one who's trying to test the medication, and maybe I made the medication, I want to see the results, I would expect results from one group over another group, and that may affect the way I collect my data. If I'm collecting the data and I don't know who got what, then that keeps me honest. And again, both of these methods are used to avoid the placebo effect. If you don't know, the placebo effect is the idea that if you're taking a medication that happens to be a placebo, a lot of times you trick your brain behaving a certain way based on your expectations. So this is just ways to cancel that out. One of the methods we can use is called blocking. So when you block an experiment, what that does is you take similar groups and put them together, right? So if I'm trying to see the effect of, like in this example, a keyboard on a certain type of phone, right? I know phones don't really use. There may be a better example for today would be iPhone users versus Android users. And then I can assign some of the iPhone users to get one keyboard or one app, some of them to get the other app, compare what they did, and get the results. And then for the Android users, do the same thing, randomly assign them to one or the other. So my blocks would be the iPhone users and the Android users, or in the original example, smartphone versus non-smartphone example. All right. One semi-common experimental practice is called matched pairs. So when you do a matched pairs design, what you're doing is you're randomizing, it's a randomized blocked experiment. So it's very similar in design to a blocked experiment. But each block is just a pair of similar units. So it's literally just like a pair. So if you can imagine up here, if we were doing something like iPhone, like a pair of iPhone users, a pair of Android users, a pair of other phones. Maybe, okay, so like iPhone, Samsung, they really do kind of have a monopoly on the market, don't they? Whatever. You get the idea. Just a pair of like similar experimental units. So that is an example of a matched pair design. You can also have a matched pair design where the order of what treatment you receive is the thing that's randomly given, right? So if you can have a matched pair that's a single individual, maybe sometimes an individual will get treatment A and then treatment B, and then the next time around, the next one would get treatment B and then treatment A, right? So the order of the treatments, if you think that might have an effect, that's a way you can. Now, making an inference from an experiment. In an experiment, we determine its effects based on whether or not it is statistically significant. A statistically significant association in data from a well-designed experiment does imply causation. So that's a big thing. In statistics, we can actually imply causation if our experiment is good enough. That's a big deal. Because everyone always says, well, correlation doesn't imply causation. You can get to causation if your experiment is designed well enough. Let's figure out how to do that. So, what kind of inference can we make? So we can either make an inference about an entire population, or we can make an inference about cause and effect. In a perfect world, we would be able to do both. If we were able to randomly select individuals and randomly assign the individuals to groups, then we could make inferences about the entire population when it comes to cause and effect. If you're only able to randomly assign individuals to groups, then you can test for cause and effect for your treatment, but that doesn't necessarily mean you'll be able to make an inference about the entire population. Now, this is usually what happens with experiments. Most experiments, you cannot randomly select individuals and perform experiments on them. That's illegal. You can do it to, plants and stuff, but usually when it comes to subjects, when it comes to people, they have to volunteer. And as soon as they volunteer, it is no longer random. Now, with an observational study, we do get to select people and not have them volunteer, which is nice. But because of that, we can't really assign them to any group and perform some treatment on them, which means we can make inferences about the entire population, but we're not going to be able to make inferences about cause and effect. So in general, usually what happens is an experiment can get you to cause and effect, and observational study can get you to an entire population. It is very rare that you're able to get both these two conditions in the same experiment. It is very rare that you're able to randomly select experimental units and then perform experiments on them, at least for anything interesting, right? You can do that to inanimate objects, but that's not an interesting experiment now, is it? Not for statistics, anyway. Now, like we said, sometimes, most of the time, like our reference, it's not ethical or practical to do an experiment. A lot of times, when we can't do an experiment, we can only do an observational study. These are the criteria. If we fulfill, we can still get causation, right? So if we are able to get all of these in an observational study, then a lot of times we can, in fact, show causation. So first of all, the association has to be strong, right? So when we're doing an observational study, we can show there's an association between X and Y. If that association is a very strong association, that's a good sign. If the association is consistent, that's also a good sign. So if it's very strong and there's not any outliers, like at all, and it doesn't maybe spread out more the further along you go, that's a good sign. When we get into really large values of the explanatory variable, we still have those very strong associations with the response variable. That's another good sign. Now we're knocking on the door. We might get some causation here. This one is really important. The alleged cause has to actually precede the effect in time. So you have to actually have a time effect, right? This seems basic, but if you're trying to show that X causes Y, then it's really important that you can show X happened before Y happened. If they happened at the same time, or if Y happened before X happened in some cases, then that's not a good look. And last but not least, the alleged cause is a plausible thing, right? So we looked at those weird associations. One of the things that came up was how many Nicolas Cage films came out in a year, correlated very strongly with how many people drowned that year. That's not a plausible causation. It does not make sense that how many films Nicolas Cage acted in would have an actual tangible effect on how many people drowned that year. That's a ridiculous thing to say. So if you have a situation like that, even if all four of these other things are true, this fifth one is a big kicker. But one example where this comes up a lot is when you're studying the effects of carbon dioxide gas on the environment. CO2 going into the atmosphere precedes the effects, the climate effects. It is a plausible effect because that relationship has been tested. The association is very strong. It's a very consistent association, and very large values of CO2 in our atmosphere do contribute to very high temperatures in our atmosphere beyond what we've seen in all recorded history so far. So those very large values associate. So this is one of those instances. It's unethical to perform an experiment with the globe to see if the globe gets too warm whenever we have too much carbon dioxide gas. But because we have a whole lot of data on that fills all five of these categories, that's why many scientists are able to say there is a causal relationship between CO2 gas and global warming, if you will, climate change. So, last, ethics. This is a big thing. It's important to be ethical. So, all planned studies must be reviewed in advance by an institutional review board charged with protecting the safety and well-being of the subjects. If you are going to perform any experiment of any kind, even if it is just a survey, you have to literally get certified to do so. I had to do this in college. It's really easy to do it as long as you're not actually giving any real treatments. Your treatments can be what kind of question you ask. That's a treatment. If that's the baseline of it, you can get certified to do that very easily. But you still have to get certified to do that legally, if we're being honest. Otherwise, if you find something cool, you won't be able to publish it. All individuals who are subjects in the study must give their informed consent before data is collected. If you don't do this, this is literally a crime against humanity in some cases. Last but not least, all individual data must be kept confidential. You can give data out for a large group, so your results don't have to stay confidential, but your individuals, your subjects in your experiments, their information has to be kept confidential. So there you go. That's data ethics. And that is experimental design. Hope this was helpful.